Hello. Hello. Listen.
जी डॉक्टर महक, डॉक्टर पूनम, पल्लवी, मानसी, oh you are waiting, I think still people are joining. Dr. Mahak, can you hear me?
Yes, sir. Good morning.
Good morning, Dr. Mahak. How are you?
Sir, I'm fine. What about you?
Good. So, how do you feel this workshop?
So, though this is not my area of research, but obviously there is so much to learn, and I think Dr. Chikindas is also able to deliver his lectures very nicely, and we are able to get whatever he wants to deliver.
I think he is putting his maximum to give the content.
Yes, sir.
And
Yes.
I'm also sharing his PPT as such to the participants.
That's great. And sir, one more. We have a query. What would be the pattern of questions for examination?
It will be descriptive type writing. So participants are expected to write a kind of write-up assignment. They have to submit within 1 hour on 25th.
Okay.
So they will be given a topic based on the lecture only, and they has to write a assignment for 1 hour with their own handwriting with their views, and they send to coordinator by email, and that will be evaluated and grading will be given. It's a kind of assignment. Is any other feedback? Any student want to ask or suggest anything? Poonam, can you hear me?
Yes, sir. Good morning, sir.
Good morning. What is your opinion? How this course is? Your feedback?
सर काफी पॉजिटिव है, इंटरेस्टिंग है. जो नॉलेज हमने ड्यूरिंग पीएचडी भी गेन नहीं की, पब्लिकेशंस स्टडी करके भी नहीं किया था, वो नॉलेज हम शेयर कर पा रहे हैं सर यहाँ पे. चिकिंदा सर काफी एडवांस नॉलेज हमें प्रोवाइड कर पा रहे हैं.
तो अभी तक हमने 2 ही दिन किया है, करीब 4 लेक्चर ही हुए हैं. और अभी हमारे पास अगले 6 लेक्चर हैं, 10 लेक्चर लगातार हैं इनका। तो मेरे ख्याल से वो बच्चे भी जो प्रोबायोटिक्स पे नहीं काम कर, जैसे डॉक्टर महक कर रही है, जिनका डायरेक्ट लिंक है वो तो है ही है, जिनका नहीं है वो भी टूल टेक्निक को समझ रहे हैं और थोड़ा सा अवेयरनेस उनकी बढ़ेगी इस चीज को कि एक्सपेरिमेंट कैसे किया जाए, टेक्निक कैसे सीखा जाए, चाहे वो एंजाइम में है, चाहे सेल ग्रोथ में है, चाहे प्रोबायोटिक्स में है, इन ऑल फील्ड दे कैन लर्न दिस। सो डोंट बी नैरो माइंडेड दैट ये तो प्रोबायोटिक की कॉन्फ्रेंस है, इसमें हमारा क्या लेना देना, ऐसा नहीं है। इस टेक्निक को जैसे हम बता रहे थे, प्रोबायोटिक्स इन ह्यूमन हेल्थ, probiotics in agriculture. So you are learning how human health is modulated. You are learning how agriculture is affected. You are learning how animal health is affected, how baby and mother and microbiota of different parts of body, how they play a role in health. So that should be taken in a very broad area, multidisciplinary approach. Anyone want to say about the course, your suggestion or anything you want to comment that will be included to improve the content of course in next 3 days lecture we have, including at least 4 days if you include today. We have completed 2 days with the 4 lectures and still 6 lectures are pending. So any suggestions, any comments that will be invited. Still participants are joining. Nitika, Arsh, Pratiksha. Pratiksha, from where you are? Pratiksha Wankhede.
Good morning, sir.
Can you hear me?
Yes, sir.
Good
Am I
morning.
audible? Good morning.
Yes, you are audible.
I am from Maharashtra, sir. North Maharashtra University, Kerala.
So briefly tell about the course and your view of its commenting. What is your view? Do you have any suggestion to give to improve the course?
Yes, sir. All courses are well, बहुत अच्छे से हो रहा है, sir.
Good.
बहुत हेल्पफुल है. हमारी PhD के टॉपिक से रिलेटेड है तो बहुत ही मुझे वो हेल्पफुल लग रहा है.
What is your topic, title of your PhD?
Exploring the prebiotic material.
Great. So you have a lecture on prebiotics also.
Yes, sir.
Yes. So any of your PhD query you can post on the chat box while delivering lecture by Professor Chikindas on prebiotics, and he will be happy to answer to solve the questions. Anyone want to say anything? Since the participants are still in joining mode, so we can interact. Any suggestion of the participant or any comment to improve the course? No one want to say? Is Gulshan? Can you hear me? Gulshan has joined from G9 lab. Gulshan, am I audible to you? No. Manjeet? आवाज आ रही है मेरी? कोई भी बता दे।
Yes, sir. Yes, sir.
All right. Okay, then let them join. We are starting. Today's tutorial will be distributed in 3 components. Component 1, we are going to discuss some of the past back analysis of Professor Chikinta's lecture study. And component 2 will be live demonstration of laboratory on probiotics. And component 3 is very interesting of industrial virtual visit. We are taking you on virtual mode to Yakult industry, which is completely based on the probiotic preparation. I hope you might be knowing Yakult. Someone has tasted, someone might be using in regular basis. Some of the students have already visited this Yakult. We are very nearby in Sonipat. So they kindly agreed to demonstrate their laboratory visit, how these probiotic preparations can be done in the large scale and how they can be utilized for commercial application. So we are having 3 components. One will go for the tutorial based on previous lecture, and then demonstration of probiotics laboratory in Department of Genetics here, and another in the industrial visit. So it's going to be very interesting. I think you will like it. Okay. As far as the previous lecture is concerned, if you have any doubt, anything we can discuss. What Dr. Chikintas delivered a lecture on the vaginal microflora and oral microflora. I mean, both components are very important for disease dysbiosis and balance. There are several level of components which are available at the microbiota in the vagina. When dysbiosis takes place, that leads to a disease which is called bacterial vaginosis. And during that bacterial vaginosis, there are a lot of metabolic changes which takes place that causes lethal degradation of the skins. And the major factor during this disease is the bacterial vaginosis is the increase in the pH. When the pH is increased, that allows the growth of several pathogenic bacteria. For example, it's like a Pseudomonas, Salmonella, typhi, Staphylococcus. These grow at the high pH. Now, the role of probiotics comes that if you have this niche with the probiotics, then it reduces the pH. Once the pH is reduced, then these pathogenic bacteria are not able to grow, and these probiotics directly has produced antimicrobial compounds like lactic acid, H2O2, and bacteriocins. All are antimicrobials in nature. So when probiotics grow in this environment, it secretes these compounds like antimicrobial substances which directly kill the pathogen. On the other hand, by reducing the pH, the ecological niche for the pathogen does not support, and that causes killing of the pathogenic bacteria. So this is how Dr. Chikindas was of the opinion that if you have this one in higher bacterial vaginosis, the antibiotics are not working. Some of the antibiotics is being used for the treatment of this disease, but there is a regular reoccurrence. This disease reoccurs because of the occurrence of several multi-drug resistant bacteria, pathogenic bacteria. And presence of this multi-drug resistant bacteria actually causes the no effect of antibiotics over it. So if you are using probiotics, actually that secretes not only one antimicrobials, but several antimicrobials are secreted at the same time. What we are saying, that is maybe lactic acid, maybe hydrogen peroxides, maybe acetic acid, maybe bacteriocins. So all this in cumulative action, cumulative manner, they act to kill the target pathogens, and those target pathogens are not being killed by antibiotics. So that is very good advantage over it, that one can use this probiotic preparation as compared to antibiotics, and it can be a very good alternative to antibiotics for the treatment of this disease. This is what, and several commercial preparation of probiotics are available for treatment of bacterial vaginosis. Antacid, antican one, another is suggested RC40. So it's like a kind of awareness when we go to the clinicians. It's not only antibiotics which are available for the treatment, but there are several probiotic preparations are also there. So this was the one component of his lecture. Second component was the oral health or oral microbiota. As we understood in the lecture that there was several level of morphological behavior in the oral system takes place. Like you have our dental bones, you have you have muscles, you have a liquid, you have a blood streams, you have a pathway, you have a solid bones, you have a soft bone. So all the environments are present in the buccal cavity, and that actually facilitates the growth of diverse type of microorganisms. Several diverse type of microorganisms are found in the oral cavity, and such type of diverse ecosystem is hardly available in the body as compared to the oral microbiota. And it is reported that there are several diseases which occur in the oral health. Related diseases are something like dental caries, apical periodontitis. These diseases, if you see, these dental diseases are increasing day by day. Earlier it was not that. Once I visited this dental hospital a day, I mean small kids like 4 years, 5 years, below 10-year kids are suffering with the periodontitis and dental caries. I mean, at our time when we were kids, I don't remember our teeth were that degraded or someone's teeth were degraded at that level. But nowadays kids are also affected, and some populations are getting more affected because of this type of diseases. Now, what is the major factor behind it? What is our food habit? Does our food habit is changing from the past to now? And that food habit actually leads to the more sensitive for the diseases, dental caries or periodontal diseases. What Dr. Chikindas suggested a study that mostly 70 to 80% population in US or somewhere they are suffering with the periodontal diseases, right? So 70 to 80% is very high number, and that has increased from the past to the now. See what kind of critical situation has come, that the population almost suffering 80% or 70% is something like out of 10, 8 people are suffering with this disease. And think of that a day will come, it may go to 100%. 100% means each one of us are having that kind of issue with the disease. And trust me that there are certain probiotic preparation. I know personally Professor John Tack is from New Zealand, and his company is over it on dental application of probiotics. He has commercialized several probiotic preparation, and it is available in the market and has shown tremendous application. In one of the slides, Indas was showing that how this probiotic preparation can manage the diseases, apical periodontitis, and dental caries. Dental caries infection even can be managed, but apical periodontitis is something which is a very deep tissue infection. Apical periodontitis means something which is a deep tissue, and when it is infected, it is hard. What the clinicians have option is only to do RCT, root canal treatment, you might be knowing. So when they do the RCT, they degrade the tooth, remove it, and fill it with the artificial material. And the tooth has no sensing ability. Only it can perform the mechanical function, but not the sensing function and biological function. So, I mean, that is not that level. But if you treat with these antimicrobials or probiotics or probiotic derived preparation, that will be great for its application. So it has a diverse environment, it has a diverse function, biofilm formation inhibition, probiotics that can be used. Several experiments have been tested. So that was based on the lecture 1. Anyone want to say anything or any doubt over it? Then we can discuss, otherwise we'll move ahead. And his second lecture was something linked with this pediatric uses, that the factors which are related with the pediatric uses was something like factors, how pediatric health is affected with the probiotics. I mean, there may be several factors, some of them like gestational age and feeding method, antibiotic exposure, introduction to solid food or something like that. And the strains which has been used is the Lactobacillus recentum and Rhamnosus have been most commonly used as a preparation for the probiotics for the application in the pediatric and in the just born baby or something like that. Because in just born baby, clinicians avoid antibiotics for several reasons. So these probiotics can be helpful for the safe application for the treatment of any disorders it is there. So that is actually a link with this probiotic applications in the human. All right. So anyone want to comment over it on the previous lecture, or if it is clear to all of you, then we should move ahead. Nothing? Okay, so we should, I should take it the way that it is clear to you. Tutorial section is something which is interactive. It should come with the interaction, the idea. You should come with the idea and put it with they try to solve this matter at this platform. And this is the reason this tutorial is meant for. I mean, this is the only platform I found in the GIAN that they are allowing a tutorial section after a lecture. I mean, you have a lecture first and then tutorial to discuss the issues raised in the previous lecture. So this is my advice to all the participants that whenever you hear the lecture, listen it carefully and note down some of the points and raise this issue to discuss over a tutorial section next day. Am I audible to all?
Yes, sir, you are audible.
But you should interact. Just sitting and listening is something like you are doing. We are not doing formality that the lecture is only for just a lecture. Come up with idea, listen this carefully and discuss. Even those who are not related with this, like Gulshan, I was just asking Gulshan because he is from Human Genetics Laboratory. I wanted to know whether it is beneficial for them or not. What level? Gulshan, can you hear me? Gulshan.
Yes, sir.
So how beneficial it is? Have you listened the lecture previously or not?
Actually, sir, I was in the Lucknow workshop for the government as related to my work. So I just here and attend the lecture on the only today. And this related to my work is that this is a new area for my research actually. So in my way, how can I able to implement it in my leukemia patients, like for the nutrition and everything? So how can it be useful? This is my prime motto for this workshop. So how can I? It will open a new area for my research. So this may be useful for me in that case.
Yeah. And see, one of the section we have probiotics in immune modulation. And in human genetics, in the application in the human health, immune modulation is one of them. So be careful on that lecture and that will be very beneficial in that case if you listen it.
Okay, yes, sir. Thank you, sir.
So anyone want to say anything? All right, I think there is no comment. So, okay, taking granted that hopefully you are slowly catch with this lecture. We have finished only 2 days. 4 days lectures are still with us. So, I will move to the second phase of tutorial, which is live demonstration of instrumentation in the probiotics laboratory, in the microbial technology laboratory, the Department of Genetics. This lab is completely dedicated on isolation of probiotics, identification of probiotics, their characterization, purification, extraction of bioactive compounds from it. So USPA is organizing this virtual visit of laboratory instruments. We are today only having what kind of equipments you need if you want to work on probiotics right from the beginning. That if you don't have anything, you go for the soil, you go for the sample and collect the sample. So Puspa, are you ready or you need time?
Yes, sir, we are ready.
So okay, if you are ready, we can start in a few minutes. Once you're ready, you can start. Meanwhile, I am connected. Right. So, starting from the beginning, that you need some biosafety cabinet. We are having this in this lab where one can handle this probiotic microorganism when you isolate it. It is very essential because once you take the sample from soil or food, you have a different types of microbiota, not only probiotics, but you may have others which may be harmful. So there must be a vertical system which exhausts the pathogenic bacteria and others to the outside. So this laminar airflow is of vertical mode, means the air moves from up to down, and that causes better elimination of most of the pathogens or harmful bacteria outside the cell. And at the same time, if you handle probiotics, the contaminants will not come in this biosafety cabinet. So there are two types. One is considered to be horizontal, another is vertical. This is a vertical mode which is most recommended, that when you take it from sample collection and take it into this biosafety cabinet, it should be useful to work it. We can go for next instrument. So you have, after growing the culture, if you want to centrifuge, this is a centrifugation machine. You need at the certain point of time, because when you grow the culture of probiotics, then you need a centrifugation. And after the centrifugation, we go for it to collect the fraction, either in terms of component or in terms of cellular part you want to do it. So it's a multi-purpose instrument for DNA isolation, for cell pellet, or ammonium sulfate precipitation pellet. You can use it. So you need a centrifugation machine at the same time when you are using this biosafety cabinet. Yes, next. This is what we have, microplate reader, or you can say ELISA reader, which is a very important multifunctional component where you have a 96 well plate. This machine is equipped with a system that if you have a 96 well plate, then all this, we'll have question answer in the last. They keep posting in the chat box. We'll answer in the last. So this kind of instrument you can use, like the 96 well plate, grow each and every culture over it, and it has a dimension in the software that each cell, each well where you are isolating, where you are putting the culture, that will grow, and you can see the dimension at the same time in the 96 well plate. So if you want to do something like activity assay or something like activity unit per ml or some protein estimation assay, this microplate reader is very important for probiotic experiments applications. Either you have a component of probiotic like bacteriocins, or you have only probiotics. You can grow. You can see the inhibition range. You can do MIC, you can do MBC, effect against the target bacteria, whether the particular bacteria is being killed by probiotics or not. Then you can use this microplate reader, or single qubit spectrophotometer that can also be used. So, but single qubit is something which takes more time than the microplate reader. Microplate reader is something which is more convenient and can be used for a high throughput assay as compared to the single one. Right. So, in a biosafety room, this room has one is biosafety cabinet, centrifugation, and spectrophotometer, which is really a common need for any microbiology laboratory. So that should be required for the growth, that should be required for monitoring the growth, and that should be required for the collection of cells from the grown culture. So you need during probiotic applications. If you are growing a probiotic, you have to collect the cells. So you need to certify good quality certification at the 4-degree unit. If you want to estimate the protein concentration in the CFS or cell-free supernatant, you need this kind of spectrophotometer. Yes, can we go for next? Right. As Dr. Chikinda suggested that probiotics, when they grow, they secrete several antimicrobial compounds like a lactic acid, like H2O2, like a bacteriocins. This is the machine which is called as EPTA purification system, which has it's like a chromatography system and very useful for the separation of bioactive compounds. You have a column, you have instrument. What let's see. Sir, instrument which you are explaining is not visible to our screen. Instrument is visible. Can you see the instrument?
Visible for us.
But some of the comments in the chat box, they are saying the instrument is not visible. Please mute yourself, unmute yourself and say it is visible or not. If you are looking at the mobile, they say it is not visible. Have you shared it now? Pushpa, can you check on your desktop? This is visible or not?
Yes, sir. Yes, sir, it is visible. Some technical issue or some layout can be changed with the participants.
Yeah, so it may be that you are not looking for good. Tanu is saying it is visible. Preeti Yadav is saying it is not visible. Prasant Kumar is saying it is not visible. See, in the mobile, it may be not visible. You have to search the window which is open. If you go for desktop, only
Yes, so Prashant and Preeti, can you change your layout option?
yes.
I think you haven't changed your layout option. That's why you are not able to see the image. Please check your layout.
So for most of the participants, it is visible. So we can go ahead, right? They say now it is visible. All right. So this is the instrument which is called as EPTA purification system. This instrument is based on the chromatographic principle. And what Dr. Chikinda suggested, I would like to understand that if probiotics are secreting like bacteriocins, lactic acid and H2O2, how you know that bacteriocins are present or not? Then you have to go for this instrumentation. You have to grow the probiotics, and while growing the probiotic, this acid is secreted in the medium. And while growing this probiotic, it secreted bacteriocins also in the supernatant medium. And you have to collect this supernatant fraction. And after the collection of supernatant, you can load this after concentration to the column. It will purify based on the ion exchange chromatography. You can use ion exchange chromatography, you can use gel filtration chromatography, and that helps actually separation of component from the top to bottom. If you have several components over it, that actually goes to the separation level. So after the separation, you can do like a fraction collector. So in this system, you have a pump, and that pump is actually connected with the column. That pumps, when it runs, it takes the solvent from the reservoir. There are two reservoirs which the solvent is being sucked by the pump and it inject into a column. And when the column goes, that actually flow from top to bottom in the ion exchange chromatography system. And by doing that, you can inject the sample, and by injecting the sample, it will run over it and collect a fraction. And by collecting fraction, you can do it. So in the purification part, we are going to demonstrate you in detail. In the last day when the bacteriocin lecture will be there, the detailed purification technique will be demonstrated to the participant. Today, I'm just indicating you the importance of this instrument to purify the bioactive component of probiotic bacteria. So you can not only use directly probiotics, but you can use the product of probiotics, that is sometimes considered to the postbiotics. Nowadays a new definition has come. So if you're using probiotics as cell, that's fine. But if you're not using probiotics as such in the application, if you want to use the metabolites of probiotics like lactic acid, like H2O2, like bacteriocins, that can be purified using this system. Okay, next. So it's like a normal technique. You can show whole view of electrophoresis, PCR, all systems, not just one. So as I told you that identification is very important part of our probiotic application. That if you are having a probiotic isolated from any sample, you have to go for its proper identification whether it is exactly the probiotic bacteria or not. So first, for this you can use like in my yesterday last time lecture, we understood that there are several level of identification. Part one is the morphological, biochemical, and molecular. So we can do morphological and biochemical with this, and this molecular part is can be done with the PCR system by isolating 16S rDNA sequence. You can run this 16S rDNA sequence on the chromatography on this electrophoresis and see the particular band of amplicon of 16S and get the sequence and analyze it. So this is how one can do using one of the most reliable technique of 16S identification using PCR and electrophoresis and get your product isolated and sent for sequencing. Once sequence comes, then you can do phylogenetic analysis and understand that whether it is lactobacillus or enterococcus or lactobacillus plantarum or not. Next, so we have a microscope where you can use microscope also for morphological characterization like gram staining and all. So by doing gram staining, you can do this part on the morphological basis identification. You can do biochemical identification just by growing it. So morphology, just you can see stain it, gram staining, and identify whether they are gram positive or gram negative. Most of the lactobacilli or lactic acid bacteria are gram positive. So that can be understood with the help of this microscope. And once you have a morphologically correct that this probiotic is available in the sample, then you can isolate the DNA, amplify 16S, and do phylogenetic analysis. You will be able to understand. So this is a very important component. It looks a smaller instrument, but I found that these are very useful for identification. Once you know you are a student, if you are not landing up with the right strain, correct strain, so at the end of your experiment of 4 or 5 years of hard research work, you might be land up with the wrong strain that it was not properly identified and I am working on different strains. So such type of precautions should be taken in the beginning, that it has to pass all the critical guidelines of the identification. Protocol of the identification should be followed correctly, and that can be used for the identification first. So once it is identified, only then that kind of probiotic can be used for the application or further characterization. We will have you demonstration in next lecture that how probiotics can be grown, what are the conditions for the growth, and that can be used for laboratory experiments, what techniques we use for the growth and application of these probiotics. Okay, so can we go to next section?
Sir, just in 5 minutes, sir.
No issue. So as you are aware that the things are almost similar, we are having in the probiotic characterization and metabolite application and characterization, but it is specific for the purpose you are using. For example, you are using bacteriocins of probiotics, then the bacteriocins are a small peptide. You need a cation exchanger, cation exchange chromatography, cation exchange column. You need gel filtration column. So size of the gel filtration is different. Yes, we use one of the autoclave for the conditions to make it sterile. Whenever you make the medium, that's fine. I'll know it. Go to next section. Yes, this is a very important component where you have an incubator cycle. This provides a condition for the growth of probiotics. Most of the probiotics, what is available, they grow like between 30 to 37 degrees centigrade. And this instrument provides a condition of 37 degrees centigrade, 30 and 45 or 42, whatever you want to set. The instrument is fixed from 5 to 80. In between 5 to 80, you can use this system. And you put your sample here, and whatever you have inoculum in the medium, that will grow actually. So we go for laminar airflow to inoculate the medium with the probiotics and put probiotics in this shaker and put the condition like temperature and shaking conditions. If you put temperature 37, that's fine. Most of the probiotics enjoy to grow. That will grow there, and at 37 degrees centigrade temperature, and rotation is probably 200 RPM they use it. So it provides an incubation condition for not only a temperature but also for the shaking environment. So I mean, this you have to grow the culture at a large scale. If you want to grow at 100 ml, if you want to grow in 10 ml or 50 ml or 200 ml, different size of clamps are available where you can put the conditions in the shaker in the suppose 500 ml medium. Accordingly, you have to have that kind of slot in the shaker to put it. Can we go next? Yes, lyophilizer is one of the instrument which is important to understand the concentration of probiotic cell or its metabolite. Like this lab is mostly dedicated on bacteriocins production. So the bacteriocins are produced in a very less amount. If the amount of bacteriocin is low in the cell-free supernatant, then suppose you have a 1 liter cell-free supernatant or spent culture or grown culture. If you grow the culture, you get the supernatant after centrifugation. This spent culture can be lyophilized. This is a very good technique that after lyophilization, the water is evaporated at low temperature. It directly sublimates into air. So it becomes powderful, and the protein is not denatured. Like you can evaporate the water at 100 degrees centigrade, but the protein present, that will be denatured. Whereas in this case, you have a temperature at very low, -70. At this temperature, everything is frozen. So it's something like water is directly from solid to gas, it is evaporated, sublimation reaction takes place, and you have a dry powder. So once the supernatant is concentrated into a dry powder, that become a very good for the high activity of the CFS against the antimicrobial activity, like for testing against Microbacillus luteus or any targeted strain that can be utilized. So what is the application of this lyophilizer for probiotics? That you can make the probiotics in the powder form, number one. You can dry this cell-free supernatant into powder form that can be used, that can be stored for a longer duration. If you want to store the probiotic cells in the room temperature, you just take it and put in the 10% milk and lyophilize it. That cells can be stored for a longer duration in the pouches or at the room temperature. You don't need any refrigeration for the storage. And just for example, if you go in the market, you'll find some sachets of probiotics which are available. Those sachets are actually lyophilized form of lactobacillus. The lyophilized powder of probiotics are being sold in the market after using this machine. Actually, they lyophilize at the largest scale and they pack over it and sell in the market. So once you isolate the probiotic, characterize for its property, and its every application is clinically approved, then that can be converted into product, and that product can be utilized for clinical application, for health application, and so on. Yes, next. This is the same, like I told you, you need a BOD system. It's a kind of chamber. This time it has been set for 40 different experiments, but mostly these people do it at the 37 degrees centigrade for probiotics growth. So this is how this instrument is mostly dealing with the incubation. Please shut down the door because experiment is already running, so it may decrease the temperature. So this instrument is actually a condition providing incubation facility. So if you take the strain from the sample, you have to incubate at the appropriate temperature and the rotation condition, and this actually allows the proper growth of the probiotics in the medium, whether you are using solid medium or using liquid medium. This instrument is more designed for the solid medium growth on the plate. If you want to grow the culture on the plate, you have to use this incubator. And earlier what we had, if you want to use this in the liquid medium, then you can use that incubator shaker. So we have a media preparation component where you can have pH meter, balance, weighing balance, rotatory orbitals where you can mix the solvent. What we are using is the MRS medium for the lactobacillus. One can prepare this MRS medium by obtaining the commercial preparation, or you can mix the component of MRS to prepare it. So this is how you need some system where you can check the pH, where you can weigh the component, mix it, and autoclave it. So this machine, which is right side to me, is something which is called a sonicator, and this one is used to break the cells. Most of the probiotics nowadays, people are using lysed cell of probiotics. Sometimes they say that probiotics are harmful. So they kill the cells and use dead cells. And trust me, the dead cells of probiotics are also functioning similar to live cells. This is very strange, but we have several publication and evidences that dead cells can perform similar function like live cells. And what is the advantage? That they are non-infectious. Now it become non-infectious. So this actually instrument is of multi-purpose. And it is used mostly to break the cells. So you have a cellular component, and whenever you want to utilize, you can utilize it for different purposes. If you are breaking the cells, after breaking the cell, the inner metabolite is coming outside in the supernatant. And after breaking, if you things has come in the supernatant, that can be purified using a fractionation system of beta fractionation system. This is what is called as ultrafiltration system. So ultrafiltration system is something, the cartridges which are fitting with the specific molecular weight. If you see on the right hand side, this cartridge is fitted in this machine where a sample can be filtered to remove larger molecular weight component from the smaller molecular weight component. If you have a mixture of metabolites, for example, if you have several different sizes proteins in a mixture, this machine is very important to separate the large molecular weight protein from the small molecular proteins. This is called ultrafiltration system, which is a GE healthcare system. And this machine actually, if you see the left side, there is a container, which is, if you put the sample in it, this container is actually taking this sample, and this sample is passed through a cartridge which is right side, and this passes from one end to another end. While passing, they have different pore size. So with the help of that, what happens, that smaller proteins are coming in the permeate, right hand side to me, which is a beaker. Through this tubing, it goes in the beaker, and left side you have a sample. So left side you have a mixture of protein, and right side it is a fractionated. So left side sample we say as a retentate, and the right side one is called as permeate. So you are going to purify based on the cutoff size. For example, if I am using 10 kilodalton cartridges pore size, this machine will allow less than 10 kilodalton protein will go right side, and more than 10 kilodalton will remain on the left side in the retentate. This is how the very good system of GE Healthcare it has provided, and it is used for the separation of different proteins from the excretory level in the cell-free supernatant. Basically, in this lab, we have used as a ultrafiltration purification system, where the protein mixture of protein can be purified using this ultrafiltration system in the large scale. Through this, you can use it, and alternative of this system is one can use salt precipitation, one can use solvent precipitation, but using this system, you don't have to go for any kind of exposure of protein to the salt or solvent. So that actually sometimes denature the proteins. But here, we are mechanically separating the proteins without denaturing it. The only thing, you have to maintain some low temperature. If you use this filtration system at low temperature, number one, it pore performs better. The filtration ability is faster and better at low temperature. And number two, the protein is not able to be denatured as compared to the salt precipitation system. So these instruments are funded by Department of Science and Technology, Department of Biotechnology, Council of Scientific and Industrial Research, and Indian Council of Medical Research, University Grant Commission, Department of Science and Technology, Government of India. These funding agencies have funded all the instruments in this laboratory to work on the probiotics and their derivatives. We are mostly working on probiotic bacteria, their probiotic properties as per se, and also their bioactive component, either in terms of bacteriocins or in terms of lactic acids or in terms of pigments. All those components of probiotics, if we can use for the clinical application, food application, that will be a great alternative to the existing food preservative which is available in the market, or clinical antibiotics which are available. As you are aware that most of the food system, they are using chemicals as a preservative, and those chemical preservatives are mostly showing the toxic side effect. And when this such type of chemicals were used as a preservative, I mean, that is very injurious for the health and harmful side effects are available. And this laboratory is dedicated to prepare natural probiotic based bacteriocins as a food preservative which can be applied. And these bacteriocins not only have shown its efficacy in food safety, but also for the application in clinical settings, like several level of clinical pathogens have been targeted and found that they are inhibited by probiotic bacteriocins. Like we have tested against Salmonella typhi, Staphylococcus aureus, Listeria monocytogenes, Aeromonas, Pseudomonas. We have tested bacteriocins from different probiotic bacteria and found to be very effective against these infectious pathogens. And by doing so, one can use this antimicrobial bacteriocins for the treatment of several infectious diseases, in contrast to the other antibiotics. Most of the antibiotics which are available in the market, they sooner or later develop resistance against target bacteria. But there are other components which are bacteriocins. Bugs are not able to develop resistance against bacteriocins because of its mode of action is different. So this is how the lab is completely dedicated for isolation of probiotics, identification of probiotics, and its characterization for human health and food. So with all these, this was the triple demonstration of laboratory which is dedicated for the probiotic application and isolation. If you have any question, anything you want to interact, I will be most welcome to answer it. If you cannot interact, you can chat, you can write your question in the text box. All right, there is no question. Oh my God. Anyone want to comment? If you want to work in this field, or if you are working in another field, these instruments can be used. No one want to say anything? Come on, interact. Interact, ask questions. That will be good for the course. Otherwise, we are just having it. Okay, this might be knowing that everything is clear to all of you, but at some time, doubt leads to a good discovery. If you don't have doubt, you don't have questions, you cannot get clarity. Come on, ask any question, anything, just your comment. If not question, then comment. Alright, I'm giving you 5 minutes. I can't see his question. He just asked, Prashant, is there a facility for processing the sample of other lab? Or we do not have custom facility of this lab, but we'll be happy to help you if you want to work with us anytime, no problem. The custom facility is available with the microbiology department. Yesterday I saw you. They have almost similar facility. If you want to use a custom facility basis, you can go there and pay them and they will do their professional. Our lab is for personal work we are doing, but the instruments are open. Anyone can learn, anyone can come and want to work with us. That will be a great. If you want any help from out of the instruments what we have in our laboratory, we'll be happy to help you. But we are not doing professional like on payment basis sample processing. For that you have to contact with some of the centers we are having in Delhi University, we are having in GNU Delhi, we are having Punjab University, we are having in Banastali also. But they do it on paid basis, I don't know. So these centers are actually dedicated to work over it. Anyone else? So it is very important that if you have an instrument, only then you can use, you can perform the experiment. The instruments are very important. Without instrument, you cannot work on it. You can unmute and ask the question. to say anything? Any one of you want your suggestion or I can't see anything in the chat box. Manoj, you want to say anything? Manoj has worked on bacteriocins mode of action study and he is here to say something. If you want to say anything, Manoj.
Sir, because you are demonstrated very well and my lab mates are. Hello? I am audible, sir?
Yes, Manoj, you are audible. Go ahead.
Sir, these techniques are very useful in the field of probiotics and isolation of lactic acid bacteria from natural resources like fermented foods and other soil samples. And the ectoprime are very useful in the purification of bacteriocin. In ectopurification system, multiple chromatography techniques which we used, like cation exchange chromatography, gel filtration, and affinity chromatography also which we run on this system. And also the most important instrument used in this purification, the ultrafiltration system. The ultrafiltration system is like a high throughput because one conventional method for precipitation of bacteriocin is ammonium sulfate precipitation. But in this method, the major problem is salt because the 90% or 80% saturation level are found during this precipitation method. But in ultrafiltration, no salt are involved. Therefore, the ultrafiltration is very useful in this purification method, and also the other instruments like my, uh, microtiter plate readers and uh, spectrophotometer, and one most important thing, centrifugation, because the self supernatant was collected using a centrifugation system. And also other instruments like incubation, lyophilization are very useful in this field for characterization, purification, and the other applications of bacteriocin. That's all, sir. Sir, you are not audible, sir. Sorry, sir. Hello, sir. Yes, sir. Yes, sir.
Sorry. Hope it is audible.
Yes, sir. Yes, sir. You are audible.
So, you told that these instruments are directly or indirectly involved in purification. One of the students is checking antimicrobial activity of bacillus. What's he asking? One of the students is checking antimicrobial activity of bacillus species against E. coli, staph and salmonella through well diffusion method, but not getting inhibitory zone. Kindly suggest. See, you have to have a control. Malasri is asking this, Dr. Malasri, that if zone of inhibition is not coming, Manoj, what do you suggest?
Sir, first of all, check the production of bacteriocin, time of production, like different time intervals, and select one indicator states which are more sensitive. After that, use other strains like Staphylococcus aureus and S. type because these are the very pathogenic, and other thing is these are the like S. type are gram negative and E. coli also gram negative. Therefore, the gram negative inhibition and gram positive inhibition are very different things. So, first of all, determine your antimicrobial activity against any sensitive and gram positive bacteria. After that, determine the other indicator strains.
So this is what Dr. Malasiri Manoj is suggesting that number one, we should go for sensitive strain, not directly on this pathogen. The main advantage of using this sensitive strain like an indicator strain, go for more sensitive like a Micrococcus luteus. Micrococcus luteus is one of the most sensitive against the condition of lactobacilli or lactic acid bacteria, and then you ensure that the growth is optimized, and under that growth condition the metabolites are secreted or not. So for example, bacteriocins are secretory in nature. When you grow, you are taking supernatant, and if the growth is not properly optimized, the component will not be secreted in the cell-free supernatant. Then you are having a cell-free supernatant without bacteriocin. If you test, you will not get the zone of inhibition. Number one. Number two, like that, if you are using this kind of strain, first you have to test on the indicator strain, which is called sensitive. You know it is confirmed, just like a control. If you take a Staphylococcus aureus E. coli, that doesn't matter, you take at the same time. But similarly, at the same time, the control should be taken like a sensitive strain of indicator strain. I would add to the Manoj point that you should check the pH also. If you are taking the supernatant, putting in the well, just check the pH. If pH is going down, it suggests that the growth is there. But after the growth, you can compare whether it is inhibitory to indicator strain and not Staphylococcus, or only Staphylococcus. So, I mean, if suppose Micrococcus is getting inhibited and Salmonella is not inhibited, it means the probiotic is producing bacteriocin, which is inhibited to Micrococcus, but it is not inhibited to Salmonella. It happens, mostly we understood that the CFS will kill all the bacteria. No, it is not possible. Sometimes one probiotic secrete a bacteriocin which kill a set of bacteria. Other probiotic secrete bacteriocin which kill other set of bacteria. No, so we have to see this kind of host range to understand how many microorganisms are being killed by this set and how many of these killed by this probiotics. So I mean, this is the beauty of this system, that you don't have one bacteriocin, so you can personalize the formulations to kill a specific target bacteria. So for a specific target bacterium, you have a specific bacteriocins, and for other you have a separate bacteriocin. This is what the specialized target can be used for inhibition. I hope it is clear, Dr. Malasiri.
Sir, sorry, sir, you are not audible, sir.
I'm audible?
Yes, sir.
So, like in most of the cases, you test the antimicrobial activity against the indicator bacteria, like zone of inhibition. But Manoj has developed a technique which is unconventional and and modern technique which has been used for the detection of antimicrobial compound, which is called as colorimetric assay. Manoj, can you highlight that assay?
Yes, sir. The colorimetric assay are totally indicator independent method. In this method, we are synthesized vesicles which were synthesized using phospholipid and fatty acid combination using sonication. And when the vesicles were synthesized, so the dark blue color, and after the interaction with bacteriocin, the color of vesicles were turned into pink or red. The pink or red depend on the nature of interaction of bacteriocin with vesicles. So bacteriocin, which bacteriocins are interact with maximum level on the vesicles, they show the dark red color. But those bacteriocins were not properly interact with the vesicles, they show the pink color. The color transition from blue to red showed the interaction and presence of bacteriocin. So in this method, there are not found any resistant and sensitive metals like other indicator stains, and these are the high throughput because within 5 minutes the color of the vesicles were turned into blue to pink, and within 1 microtetraplate or 96 pill plate you can screen one at one time 96 bacteriocins using microtetraplate and using this method. So this is the very fast and high throughput method for screening of bacteriocins from natural resources.
Yes, so it is a very important and relevant technique where you are using this kind of, mostly people are using conventional method, but this is indicator independent. It's just like you use vesicle as a target and take a bacteriocin. When you target the bacteriocin with the vesicle converts its color from blue to pink, what Manoj is suggesting. And within a fraction of seconds, you can get the detection of bacteriocin. Otherwise, if you are doing antimicrobial activity based on zone of inhibition, it takes like a 24 hours. More than 24 hours, it depends on the indicator strain. But in this case, we are just taking the vesicles and we can go for it. Testing within no time. So it's a specific, faster as compared to the zone of inhibition. All right. Okay, then I think this demonstration part is completed, and now we are waiting for virtual visit to our factory Yakult, and this much is, it may be interesting for the participants' benefit that if you produce the probiotics in the lab and if you go for industrial visit, you can see the real application of this product, how this can be applied. So meanwhile, the industry personnel is joining, you can wait. Just wait, within a few few minutes, within 5 minutes, they will join.
Good afternoon, sir.
Yes, Sanya, good afternoon. Are you here?
Sir, I'm from Yakult. So, shall we begin with the virtual factory tour?
Yes, we are waiting for you. Thank you very much for joining.
Thank you, sir. So, I'm sharing my screen. So, why don't you please confirm if it's visible to you?
Right. So I'm very thankful to Dr. Neza Hazela and the entire team of Yakult in India Private Limited for their consent that they organize this virtual tour for our participant, which will be highly beneficial because I found this company is directly connected with the application of probiotics, and that will be helpful. Sanya, it is visible to us. You can go ahead.
Thank you so much. So, firstly, on the behalf of Yakult and on India Private Limited, I welcome you all here to the virtual factory tour of Yakult. So, before starting with the presentation, it's my request to you all, please mute your mic, and it's my request, kindly do not record the video. So, today we are going to discuss about history of probiotics. Then we need to discuss about what is Yakult. After that, global presence and science behind Yakult. And then lastly, we will be discussing about the distribution channel of Yakult in India. So as of now, talking about history and evolution of probiotics, you all can see this picture. He is Dr. Eli Mechnikov, the name and reason behind the discovery of probiotics in the world. So basically, he was a research scholar, and the area where he was doing his researches, that was a village near Bulgaria. So what he concluded that people living in Bulgaria, and especially farmers, they were having a very healthy lifestyle. But at that time, due to lack of technology and evidences, he was not able to find the exact reason which is responsible for maintenance of good health. So he just came up with this concept of longevity without aging into a book which was known as the prolongation of life. And for that particular book, he was also awarded Nobel Prize in 1908. So after that, when many scientists came and they started studying about role of several kinds of bacteria on human health, so among them there was a scientist who was known as Dr. Minoru Shirota. So he became the first scientist in world to isolate and culture the particular type of strain which we are using in Yakult, and this is known as Lactobacillus casei strain Shirota. So in 1930, this Lcs bacteria was isolated, and in 1935, the first bottle of Yakult was launched into Japanese market. So now talking about probiotics, that what are probiotics in actual? So you must have heard about these two terms, FAO and WHO, which is Food and Agriculture Organization and World Health Organization. So basically these are the two UN organizations which came up with the definition of probiotics in 2001, and what they said that these are the live microorganisms which when administered in adequate amount confers health benefit on the host, which means that any type of microorganism, if they are reaching live in your intestine and if they are providing you with some sort of health benefit, so that particular type of food will be termed as probiotic food. Now let us see what is the best time to drink Yakult.
How's your coat, man? What time of day should I drink your coat?
Because Yakult is just like other food, you can have it at any time of the day. But drinking it every day is what's important, so make sure it's at a good time for you. Drink Yakult at a time of day that's best for you.
So basically basically you can have Yakult at any time of the day, whatever your body prefers. So now the philosophy on which the business of Yakult is based, so it is known as Shirotism. So Shirotism basically deals with the three philosophies. The first one, prevention is better than cure. Second one, a healthy intestine leads to a long life. And the third one is affordable price. Because at that time when Yakult was launched into the Japanese market, so that was the time of World War. And people were not that much financially stable, and Japan was a very poor country. So they introduced Yakult at the price of postcard so that everybody can afford it easily. Now talking about the products which we have, so we have these two products, Yakult Probiotic Fermented Milk Drink, a 65 ml bottle which consists of 6.5 billion of live bacteria made up of without any type of preservatives, stabilizers or coloring agents. Talking about its ingredient, it has water, sugar, SMP, natural and nature identical flavor plus the bacteria. So the price is rupees 80 for 5 bottle pack in case of Yakult Original, and it's rupees 95 in case of Yakult Light. The best before or expiry date, which we call as, it's 40 days from the date of manufacturing. And when we talk about storage conditions, so as we all know that it's a bacterial product, so it needs a particular type of refrigeration. So it is 40 days from the date of manufacturing. Along with it needs to be kept below 4 degree, which means that the normal refrigeration temperature is required, but at max it can be kept below 10 degrees Celsius. So now talking about the benefits of Yakult, so it not only helps in improving digestion, but yes, at the same time it helps in building immunity also. So this drink has been scientifically proven with more than 80 years of scientific research, which suggests that above the age of 1 year, anyone can drink Yakult without any type of side effects related to it. Now the next question is, can we heat up Yakult?
Tell us, your coat man. Can we heat up your coat?
Yakult should always be stored below 10 degrees Celsius. High temperatures can cause the lactic acid bacteria in Yakult to weaken and even die. And although short periods are okay, if Yakult is left out too long, the quality can suffer. So do be careful. Also, using a microwave or direct heat can damage the container, so please don't do it. Please don't heat your Yakult.
So yes, we cannot heat up Yakult because the bacteria which is inside this particular probiotic drink, it requires a particular type of temperature. So if we are providing the above temperature to it, so it will not be able to survive and the number starts decreasing. So it's essential to maintain that particular type of temperature. Now, these are few of the safety certifications which have been granted to us related to bacterial safety. So we have this GRAS from US FDA, which is generally recognized as safe from US Food and Drug Administration. After that, we have this FSSAI product approval, and then it's the FOSHU label, which is food for specified health uses. Now the science behind Yakult. So yes, there are more than 100 human studies that have already been conducted on this particular strain, and you can see these two pictures. So these are the Yakult State of Art Research Institute. Basically, the research and developmental centers of Yakult, where different type of researches are going on related to bacterial safety. So the one is located in Japan, and the another one is in Belgium, which is in Europe, where more than 200 scientists are regularly working on improving the quality and efficacy of this particular strain. Now let us see what is the science behind Yakult.
Our gut is home to trillions of bacteria that are good and bad. For a state of good health, the beneficial bacteria must outnumber the harmful disease-causing ones. The beneficial bacteria or the friendly bacteria are called probiotics. In other words, they are live microorganisms which, when administered in adequate amounts, confer a health benefit on the host. Probiotics aid in digestion and promote better absorption of nutrients. They are available as specially formulated dairy foods that contain billions of good bacteria. And because they are easy to administer, many medical experts use it either for therapy or preventive purposes.
We have the acute self-limiting diarrheas, where antibiotics unfortunately are so frequently misused in this country. I think a safer alternative would be to use probiotics.
So what does probiotics mean to India, where diarrhea kills tens of thousands of children every year? To find out its efficacy in preventing diarrhea, the National Institute of Cholera and Enteric Diseases in Kolkata undertook a study.
We selected about 4,000 kids less than 5 years of age, and we gave 2,000 of them the probiotic drink, which contained Lactobacillus casei strain Shirota. And the other 2,000 kids, we gave a nutrient drink. At the end of 6 months, there was a 14% reduction in diarrheal incidence among the children who had received the probiotics.
Excuse me, sir. Before starting with the presentation, it was announced that you're not allowed to record this particular session. So it's my request to you, please don't record it. After that, I'll start with this particular video. Because it's against our company's policy, so you cannot record this particular session.
Right, it is already done on recording mode. So recording mode is removed for a while. You can present.
Okay, sir. Okay, thank you.
Who had received the probiotic drink.
Probiotics are seen to benefit in many gastrointestinal disorders.
I find that they have sometimes almost magical effects, and people often wonder, what will really make the difference to them.
My name is Naina. I'm 5 and a half.
Till a year ago, Naina used to suffer from incessant GI disorders until she was introduced to Yakult, a probiotic drink to be taken on a daily basis.
Using Yakult, the frequency of some infections has really gone down.
I like Yakult because it's milk, but I don't like milk. I like Yakult better.
Yakult is a popular probiotic milk drink that contains more than 6.5 billion Lactobacillus casei strain Shirota. Way back in 1930, this strain was discovered by Dr. Minoru Shirota, a Japanese scientist who isolated and cultured it. Later, he used the strain to make Yakult.
The Ministry of Health, Labour and Welfare in Japan has granted Yakult the 4-shoe label. That is a food for specified health uses label, and this label is given to only those products which are scientifically proven.
Today, Yakult is a popular probiotic drink in Japan, and with more than 30 million people drinking it every day in 32 countries, it's the most credible probiotic drink available.
Since we were looking for a product that is scientifically validated, we started using Yakult in our hospital. We did not have too much of the fresh live bacteria available, but now when we see there are products which are available, like you have this Yakult, which is in the live form, it's like a wonder drink.
Currently, I'm using Yakult because I think it's a nice drink which people accept well.
As more and more findings emerge, it won't be long before the multiple uses of probiotics percolates within the medical fraternity. But one thing's for sure, probiotics are here to stay to give us all a good gut feeling.
So now talking about the business part of Yakult in India. So Yakult Danone India Private Limited is a 50-50 joint venture between Yakult Honsha of Japan and Danone of France. So it was launched in Delhi NCR in 2008. After that, we have covered every major city and state. The total investment on this plant right now is of more than rupees 360 crores, and the state-of-art production facility is located at Rai, Sonipat, Haryana, with a production capacity of 10 lakh bottles per day. So this is how our factory looks like. This is the outside view of our factory, and whole factory has been designed by following all the ISO standard. So this production state-of-art facility is located at Rai. Now these are few of the pictures where we have depicted the process of Yakult, that how in an actual manner the process takes place. So the first picture is of the seed room. Actually seed is known as the starter culture. As we know that to initiate fermentation we need to have a starter culture first. So this is the particular area where the seed or the starter culture is prepared. We have 3 tanks inside here, and inside these tanks the seed is prepared. After that you can see the other machineries as well, that in how hygienic and systematic condition the Yakult is being prepared. And then it's the homogenizer machine. The function of this particular homogenizer machine is to homogenize this particular liquid into the even particles so that it can be formed in a easily drinkable pattern. So if any kind of lump is formed during the fermentation, it breaks down that particular lump and create a uniform pattern for the dilution. Now the second one is the IBM section, which is the bottle manufacturing section. So these both machineries which you can see on the screen, so they have directly been brought up from Japan, and our whole factory is automated. There is as much as manual touch as we can do. So the empty bottles of Yakult are also being prepared here only so that we can reduce the scope of contamination. So they have been directly brought up from Japan, and they have the highest outrated capacity, which is 11,000 bottles per hour by a single machine. So we have 2 of them, which means 22,000 bottles in an hour can be prepared here easily. Now this is the filling and packaging section of Yakult. So the first picture is of the unscrambler machine. So the function of this machine is to assemble the bottles in an upright direction so that it can be filled easily. After that filling takes place, and after the filling, immediate capping of bottle is done, because we need to reduce the scope of contamination as well, plus the bacteria which we are providing in this particular Yakult, which is the LCS bacteria, it is anaerobic in nature. So it cannot survive in the presence of oxygen. So that is the reason that we are immediately doing the capping after filling. So now the next is quality control lab. So this is basically the area where the quality parameters checking take place. Every kind of biochemical or physiochemical testing takes place here, and there are around 120 quality parameters which are tested, and also the raw material is being tested here. Once they qualify that this particular drink is safe to use and it contains this much amount of bacteria, only then the product is transferred towards the packaging section. So there is a small video related to factory operation. Here we are showing that starting from the seed room, like preparation of starter culture, up to the packaging and logistic part, how everything takes place. So please have a look towards this video.
Hello everyone, welcome to the virtual show of Yakult. It is always the growing state of the art unit, home to treatment of virtual activity. Our factory is located at Sonipat, Haryana. The area of factory is spread over 8 acres, out of which 30% is green area. The factory is designed as per the international standards, and special care is taken to maintain highest level of hygiene while making Yakult. Our factory is reputed with ISO 9001 for quality management system, HACCP for food safety and OSAS for human safety certificates. Yakult is very much conscious for clean energy, and we have solar plant with a capacity of 742.5 kilowatt peak. Yakult is a Japan-based company, and worldwide we are present in 40 countries and regions, and over 40 million Yakult bottles run every day. Way back in 1930, this gene was discovered by Dr. Minoru Shibata, a Japanese scientist who isolated and cultured it. Later, he used this gene to make Yakult. Localization of Yakult started from Taiwan in 1964, and today we are in 40 countries and regions. Okay, so now I'll show you a presentation. Regularly, many visitors come to see our production facility and witness automated process. Process of manufacturing Yakult has various stages of operations. Remember, all equipments and area around them are carefully sanitized and sterilized before the use. We have capacity to produce 1 million bottles in a day. Now, moving to the production activity. The first stage is making of seed starter. We take skim milk powder, glucose, and mix it with the hot water and sterilize with the steam. Then we cool down the solution and add our Lactobacillus casei enchirota in the seed tank. This culture then starts to ferment and seed starter is prepared. In culture and mixing room, we prepared thousands of liters Yakult. Once we complete fermentation process, we add sugar solution in it and send it to the blending section to mix it with the chilled water. This is our quality control room. Here all the quality checks are done right from the raw material to the finished product, where all the microbiological parameters of our product are continuously monitored and controlled. Now Yakult is quality checked. Now it is time for moving to the further operations. This is our injection blow molding room. We have 2 injection blow molding machines to manufacture empty bottles. The capacity of each machine is 11,000 bottles per hour. Our bottle is made up of food grade material. This material comes through vacuum pipe and enters the machine through the hopper. The bottles are coming out of the machine and transferred into the bottle storage tank. Through clean air pressure, we can look through this glass to see how it works. Now we are in the filling machine area. The bottles falls from the bottle storage tank into the unscambler in random fashion. Now because we need bottles in upright position for filling delicious Yakult, the unscambler machine helps in doing that. Now just before the filling machine, clean bottles passes through the printing machine, where logo and important information are printed. Then printed bottles enters into the filling machine, where our invoice number 1 probiotic treat Yakult is filled and capsules. At this stage, this machine fills 45,000 Yakult bottles per hour. Finally, random physical checks are done to ensure that the bottles are ready to go for packaging. These moving bottles looks like a toy train, which is fun to watch. Now bottles enters to the packaging area, where first machine packs pack of 5 bottles each, and second machine packs pack of 50 bottles each. This is the most amazing process. Then empty pallets are coming through the first conveyor. Then pack of 50 bottles are stacked on pallets and moved towards cold storage area through second conveyor. We shift stacked pallets into the cold room with the help of forklifts. Cold storage temperature is maintained in refrigerated condition under 10 degrees Celsius. Hope you all enjoyed our virtual lecture tour. See you again with friends and family members. Thank you. Thank you so much for coming.
So now I hope after watching this video you are very much clear that how in actual the Yapur processing takes place here. Now lastly discussing about the distribution channel. So we have divided it into three major parts: retail, e-commerce and home delivery. So Yakult is available in several stores and hotels, and more than 250 hotels are giving Yakult to their consumer as a part of breakfast meal. Now the e-commerce platform. So you all must have been using all these e-commerce platform: Amazon, Swiggy, Big Basket, Zepto, Blinkit. So the product is also available here online. You can also purchase from these online web stores. The home delivery channel, which is the most significant channel. So this one is handled by Yakult ladies. We call them as ambassadors of good health because they provide good health to the consumers at their doorstep in the form of Yakult. So more than 300 Yakult ladies are associated with us in India, and when we talk about world, the number is more than 80,000 of Yakult ladies. So thank you very much for attending this presentation, and now I hope that you are very much clear that what are probiotics and how they are being used in food industry to create a better health. So now if you have any question related to product, related to company, or anything which is not clear to you, you can ask. Thank you very much.
Thank you, Sanya. Thank you very much for presenting your everything from outside to inside and the mechanism of operating the system, and hopefully it is directly linked to the participants for their benefit.
Thank you, sir.
Anyone want to interact with Sanya is fine, otherwise she will leave. So Sanya, anyone want to visit if your company, what is the procedure is? Anyone can go or you need a specialized formal?
Sir, you need to send us a requesting proposal mail regarding the industrial visit, and then according to the slots which are vacant, we'll provide you the dates, because we have visits every day and every day 3 to 4 groups come here. So according to the availability, we'll provide you the slots for the students.
Suppose these participants individual want to visit, so is there any provision for individual? Or
yes, sir. Also individually you can visit. You just need to drop us a mail, or you just need to contact us through our mail IDs or contact number, and then we'll provide you the dates and the time.
Oh, very good. So this is good for the participants that whenever and wherever you are, if you are interested to visit, go to the website and there is a link. You can email to the concerned person and they reply active in this. So if there is no question, I would like to thank Sanya for all her support during the visit. This visit was very much important and related with the kind of course content we are running under this international workshop.
Thank you so much, sir. Thank you.
Thank you very much, Sanya. So, advice for the participants that we are at the end of this tutorial session. We will do it tutorial tomorrow at the same time, and we'll meet in the evening for the lecture at 5:30. So see you soon.
know